Hey guys, so in this video we're going to be going over meiosis and sexual life cycles. So we know that living organisms, we are distinguished by the ability that we have to reproduce our own kind. And offspring, they can resemble their parents more than they resemble less closely related individuals of the same species. When we have the transmission of traits from one generation to the next, it is called heredity. The differences that we have between parents and siblings, that demonstrates variation. When we are talking about genetics, we are talking about the scientific study of heredity and heredity variation. So we know that parents, they endow their offspring with coded information in the form of genes. Your genome, it's made up of the genes that you inherited from your mother and from your father. Those genes, they program specific traits that are going to emerge as we develop from fertilized eggs into adults. Genes are segments of DNA, and they are genetic information that it's going to be transmitted as specific sequences of deoxyribonucleotides in DNA. Most genes program cells to synthesize specific enzymes and other proteins whose cumulative actions produces in organisms inherited traits. When we are talking about a genome, what we are talking about is all of the chromosomes that make up our genes. So this is what we're seeing, like inside of a cell, we see our chromosomes, we have our DNA, this is a segment of DNA, this is a nucleotide, and every single gene that we find inside of a cell makes up our genome. So in plants and animals, when we're talking about reproductive cells, they are called gametes. So gametes, they are going to transmit genes from one generation to the next. After fertilization, fertilization is defined as the fusion of a sperm cell and ovum. So after fertilization, genes from both parents are going to be present in the nucleus of that fertilized egg. A fertilized egg is now called a zygote. So that's exactly what we're seeing. Here we're seeing an egg cell, and this egg cell had 23 chromosomes, and this sperm cell had 23 chromosomes. When it was fertilized, this egg and this sperm cell, they came together to form a zygote. And inside of the zygote, we now have the 23 chromosomes from the mom and 23 chromosomes from the father, meaning that this cell now has a total of 46 chromosomes. So our chromosomes, they're going to consist of a single DNA molecule that is going to be associated with a variety of proteins. Almost all DNA in a eukaryotic cell is going to be subdivided into a chromosome in the nucleus. We have very small amounts of DNA that are also found in the mitochondria and the chloroplasts. So every single living species, it has a characteristic number of chromosomes. When we're talking about humans, we have 46 chromosomes in our somatic cells, and we have 23 chromosomes in our gametes. So each chromosome, it consists of a single DNA molecule that is going to be associated with a variety of proteins. And each chromosome has hundreds or thousands of genes that is going to be at a specific location, which is called a locus, that is along the length of the cell. So in this slide, we're going to really dive into the difference between a gene, a locus, and an allele. So we already said that a gene, it is a segment of DNA that codes for a specific protein. And genes, they're also going to contribute to a particular function or trait. So this is a chromosome. It has genes everywhere, right? So like it's gonna have all of this, these are genes everywhere on this chromosome. When we are talking about the locus, we're talking about the physical location of a specific gene on a chromosome. We can think of a locus as the address on the chromosome. And that's what this is saying. The flower color gene is always going to be located right here on the chromosome. Now, we might have petal shape over here. Well, petal shape is always going to be here. So the locus for petal shape will always be in the same area. And when we get more specific and we talk about the alleles, what we're talking about is the expressed version of the gene. Is it white or is it purple in color? So in asexual reproduction, a single individual is the sole parent that is going to donate its genes to its offspring. Asexual reproduction is going to give rise to a clone, a genetically identical individual. So our single-celled eukaryotes, they can actually reproduce asexually by mitotic cell division, and they can produce two genetically identical daughter cells. An individual that can reproduce asexually, it gives rise to a clone, and members of a clone, they can be genetically different as a result of mutations. In sexual reproduction, we have two parents that are going to produce offspring that have unique combinations of inherited genes. Unlike a clone, offspring produced by sexual reproduction vary genetically from their siblings and their parents. So when we're talking about a life cycle, we're talking about generation to generation sequences of stages in the reproductive history of an organism. So by these standards, a life cycle is defined as beginning at conception, and it continues until that organism produces its own offspring. So in our cells, in humans, each somatic cell has 46 chromosomes. During mitosis, the chromosomes, they become condensed and then they're going to be visible under a light microscope. At this point, chromosomes, they can be distinguished by their size, by their centromeric position, and the pattern of staining with certain dyes. We can produce images of the 46 human chromosomes, and we can arrange them in pairs in order of size to produce something called a karyotype display. The two chromosomes in a pair have the same lengths, same centromere position, and same staining patterns. These are going to be called homologous chromosomes. In other words, homologous chromosomes, they are the ones that are found in pairs. Homologous chromosomes, they carry genes that control the same inherited characters. When we have two distinct sex chromosomes, such as our X and Y chromosomes, they are an exception to that general pattern of homologous chromosomes in human somatic cells. Our human females, they are going to have a pair of homologous chromosomes that will be XX. And males, they are going to have XY. When we are looking at this figure, we are looking at the karyotype of a normal male. So he has 46 chromosomes and he is XY. Our chromosomes, they are going to be sorted by size, centromeric position, and by staining pattern. So when we look at our homologous chromosomes, again, our homologous chromosomes are all the ones that we find in pairs. We see that their staining patterns are the same, they are the same length, and their centromeres are located at the same spot. Then we get to our two distinct sex chromosomes. Most of the genes carried on the X chromosomes, they are not found on that tiny Y chromosome. I mean, just look at how different in size our X and Y chromosomes are. They are our 23rd pair of chromosomes. So only small parts of our X and Y chromosomes are homologous, but overall, our sex chromosomes, they are not homologous, and that is because they are not found in pairs. So the other 22 pairs of chromosomes, our homologous chromosomes, they are considered, or actually, they are also called autosomes. So the occurrence of homologous pairs of chromosomes is a consequence of sexual reproduction. Remember, we're going to inherit one chromosome of each homologous pair from each parent. So we're going to have a paternal one and we're going to have a maternal one. The 46 chromosomes in each somatic cell, they are two sets of 23, a maternal set and a paternal set. And the number of chromosomes in a single set is going to be represented by N. Any cell that has two sets of chromosomes is going to be called a diploid cell. Diploid cells, they are going to be abbreviated as 2N. So anytime we have a cell that has two sets of chromosomes, it is called a diploid cell. And a diploid number of chromosomes is abbreviated as 2N. Our diploid cells are all of our somatic cells because our somatic cells all have 46 chromosomes. So our homologous chromosomes, again, they have the same genes in the same order, and the place that they vary is their alleles. So the color that our eyes express, the shape that our nose has. So our sex chromosomes, again, when we have a male, we have XY, and a sperm is going to carry either X or Y. And when we have a female, they are XX, and their egg or their ovum is always going to carry an X chromosome. When we are talking about our sex chromosomes, we are talking about haploid cells. Again, that is abbreviated as N. Our sex chromosomes, they only have one set of chromosomes. There are no pairs. So our sex chromosomes are always haploid and our somatic cells are always diploid. So when we're going to talk about chromosomal behavior in humans, we have to think about the human life cycle. Essentially, the human life cycle, it begins the moment that a haploid sperm cell fuses with an haploid ovum. And the union of these gametes, it culminates in that fusion of their nuclei, which causes fertilization. And that is what we are seeing right here. We have a sperm cell and we have an egg cell. They come together and they form a zygote. So remember, zygotes are post-fertilization. So that fertilized zygote it is now diploid because it has two haploid sets of chromosomes. So we went from having haploid gametes to now having a zygote that is diploid. It contains two haploid sets of chromosomes from the parents. Our gametes, they are going to actually develop in the gonads, and the gonads are our testes or our ovaries. Our gametes, they are not produced by mitosis. They are produced by meiosis. Our gametes develop from specialized cells called germ cells that we have in our gonads. Gametes undergo that process of meiosis, in which the chromosome numbers are halved. The human sperm or ova, they have a haploid set of 23 chromosomes, one from each homologous pair. And fertilization, it is going to restore the diploid condition. Remember, this was haploid, and the sperm was haploid. And when they came together, it became a diploid. So our organisms, they have a variety of sexual life cycles. Most of the animals, including humans, have the first type of life cycle, which is that haploid life cycle. What this means is that gametes are the only haploid cells that we have. Our gametes do not divide, but instead they're going to fuse to form a diploid zygote that divides by mitosis to produce a multicellular diploid organism. We have plants and some algae that have this secondary type of life cycle that is called alteration of generations. This means that the life cycle has two multicellular stages, a haploid and a diploid stage. And then we have another form of a life cycle, which is found in most fungi and some protists, which is a diploid life cycle. During this time, gametes fuse to form a zygote, which is the only diploid phase. That zygote is going to undergo meiosis, and that's going to produce haploid cells. So either haploid or diploid cells can divide by mitosis depending on the type of life cycle. But only diploid cells can undergo meiosis because haploid cells have a single set of chromosomes that cannot be further reduced. And even though the three types of sexual life cycle differ in the timing of meiosis and fertilization, they do share this really fundamental result of genetic variation among offspring. So it's really important that we remember that meiosis reduces the number of chromosome sets from diploid to haploid. Many steps of meiosis are going to resemble the steps of mitosis. Both meiosis and mitosis are preceded by the duplication of chromosomes. This happens in interphase. When we're talking about meiosis, we have two consecutive cell divisions. We have meiosis 1 and we have meiosis 2. This is going to result in 4 daughter cells. The first division of meiosis is going to separate homologous chromosomes. That second division is going to separate sister chromatids. And the 4 daughter cells that we have at the end of meiosis 2 have only half as many chromosomes as the original parent cell. Here we started with 4 and here we ended with 2. And when we look at them, the ones that we started with, or here we go, the ones that we started with, they are genetically different from the parent. So meiosis 1, it is preceded by interphase, in which our chromosomes are going to be duplicated to form sister chromatids. We have two genetically identical sister chromatids that are going to make up one duplicated chromosomes. Those sister chromatids, they're going to be closely associated all along their length, and this association is called sister chromatid cohesion. In contrast, the two chromosomes of a homologous pair, they are individual chromosomes that were inherited from different parents. So during prophase 1, we have our chromosomes condense. We have our homologous chromosomes pair up. This is called synapsis. When one homologous chromosome from each parent comes together, that area in which they touch is called synapsis. And because we have two homologous chromosomes pair up, they form something called tetrads. We also have crossing over occur during prophase 1. So what happens is that crossing over, it occurs as something called the chiasmata. And this is the point where our homologous chromosomes are physically going to exchange genetic material. So again, we're just going to go into more detail about the formation of our tetrads. So essentially, when we are in meiosis 1, we start off with homologous chromosomes that are next to each other. This section that they touch is called synapsis. This area of crossing over is called the chiasmata. During prophase 1, our homologous chromosomes, they pair up during synapsis. They're going to be held together by this synaptonemal complex, and each homologous pair is going to form a tetrad. So they're going to now have 4 total sister chromatids, right? Because this is a sister chromatid. This is one, and this is one. So we have a total of 4 sister chromatids. In metaphase 1, our homologous pairs, they're going to line up at the metaphase plate, and these pairs, they're going to line up randomly. Remember, homologous chromosomes are 2 duplicated chromosomes that line up next to each other. Each homologous chromosome is made of 2 sister chromatids. Therefore, we have a total of 4 sister chromatids in a homologous chromosome pair. During anaphase 1, these homologous chromosomes, they're going to be pulled apart to opposite sides. Our sister chromatids stay together because of cohesins at that centromere. So at this time, the cell wants to reduce the number of chromosomes. It goes from being diploid to haploid. It separates whole chromosomes, not chromatids. In telophase 1 and cytokinesis 1, we have that cell splitting. During telophase 1, we have these chromosomes reach the opposite sides, and then that cell is going to divide into 2 haploid cells. The nuclear membrane, it may reform around each set of chromosomes, and each new cell has half the number of chromosomes. They are now haploid. Cytokinesis means that the cytoplasm is dividing, and it is resulting in 2 haploid daughter cells. And now each new cell has 1 set of chromosomes, but each sister chromatid is still connected. So prophase and telophase, when we're talking about meiosis 2, they are basically identical to meiosis 1. Where we really differ is in metaphase. During metaphase 1, we had chromosomes line up in homologous pairs, which meant that we had 4 sister chromatids. In metaphase 2, those chromosomes, they now line up in a single row. And it also differs in cytokinesis. When we have cytokinesis 1, it's going to result in 2 non-identical haploid daughter cells, but cytokinesis 2 is going to result in 4 non-identical haploid daughter cells. This chart is summarizing what happens in meiosis 1 and what happens in meiosis 2. Feel free to pause and look it over. And this is just meiosis 1 and meiosis 2 together. And here we're comparing mitosis and meiosis. So if you ever get lost thinking what happens where in which cycle, you can refer to this chart and it has a lot of those answers. So mutations, they are the original and primary source of genetic diversity. These changes in an organism's DNA, it creates the different versions of genes, and we call those our alleles. Once different versions of genes arise through mutation, we have something called allele reshuffling during sexual reproduction that is going to result in members of species having a unique combination of traits. So our sexual life cycles, they produce genetic variation among offspring. What this means is that the behavior of chromosomes during meiosis and fertilization, it is responsible for most of the variation that arises in each generation. We have 3 mechanisms that are going to contribute to genetic variation that arises from sexual reproduction. We have independent assortment of chromosomes, we have crossing over, and we have random fertilization. When we're talking about independent assortment of chromosomes, it is going to contribute to genetic variability due to the random orientation of homologous chromosomes during meiosis 1. So essentially, independent assortment of chromosomes, it just creates genetic diversity. When we're talking about crossing over is going to produce recombinant chromosomes. Recombinant chromosomes is going to combine genes that are inherited from each parent. During meiosis in humans, we have an average of 1 to 3 crossover events occur per chromosome pair, and the amount that we have is dependent on the size of the chromosome and the position of their centromeres. So crossing over by combining DNA inherited from 2 parents into a single chromosome, it is a really important source of genetic variation. When we have metaphase 2, we have non-identical sister chromatids that sort independently from one another, and this is just further increasing that number of genetic types of daughter cells that are formed by meiosis. And the random nature of fertilization also adds to genetic variation that arises from meiosis. Think about it. Any sperm can fuse with any egg. And the ovum is one of about 8.4 million possible chromosome combinations. The successful sperm is one of about 8.4 million possibilities. So that resulting zygote, it can contain any of more than 70 trillion possible combinations. And then crossing over adds even more variation to this. So Charles Darwin, he recognized the importance of genetic variation in evolution. He said that as the environment changes, the population may survive if some members can cope effectively with these new conditions. And because mutations are the original source of different alleles, those are then mixed and matched during meiosis. When we have new and different combinations of alleles, it may work better than those that previously prevailed. So that ability of sexual reproduction to generate diversity is widely recognized as a major factor in its evolutionary success. Think, when we have a fairly stable environment, asexual reproduction, it seems to be more advantageous because it ensures that perpetuation of successful combination of alleles. Sexual reproduction, it is expensive. Its energetic costs to that organism is a lot higher than those of asexual reproduction. In sexual reproduction, we produce generally one offspring when we're talking about humans. In asexual reproduction, we can produce hundreds to thousands of offspring in one cycle. So, overall, the fact that we have genetic diversity in sexual reproduction, it allows us to better adapt to the changes in our environment. Even though asexual reproduction is more efficient, it can actually harm evolution because it does not change the same way. Those organisms, they don't change the same way that the organisms that are produced through sexual reproduction change.